Thanks, Nicholas, for having us. You've been an incredible host, and so is all your team. This is really, really awesome. I'm super excited to share my story today. Um, in 21, I was living an amazing life. 10 years before, I brought my company GitLab to the US. I did Y Combinator, I raised funding, and now 6 years later, that was actually 6 years after Y Combinator, we took it public. I was riding an enormous high, but then a year later, I had really, really bad news. One day, I was doing a bench press, and I felt a strain across my heart. And I'm like, I know this. This is not a heart attack because I had it before, and it lasts a while, and it goes away. It didn't go away. 2 weeks later, I'm still like, this is the world's longest heart attack, or there's something else. But I'm not feeling good. At 4 a.m., I got up. My wife's over there. I told her, hey, I'm just going to the emergency room at 4 a.m. Don't worry about me. And at the hospital, they said, yeah, heart attacks in the last 2 weeks. You're fine. They took an X-ray, sent me home. And then my GP called a few hours later while I was working, and he said, hey, do you know how to meditate? I'm like, wow, that's a very Bay Area question. But I fully embraced the Bay Area. So I did a silent retreat with the Buddhists once in the Santa Monica Mountains. Yes, I know how to meditate. He's like, well, you better start now because your blood pressure was 170, and the pain you're feeling might be your aorta starting to burst. So, hey, totally chill out and take the first Uber to the emergency room. I'll tell them you're coming. And there they had good news and bad news. I had no aneurysm, but I did have a 6-centimeter tumor growing out of my T6 vertebrae. So I was in a lot of pain. We immediately went for surgery, a lot of radiation, and a ton of chemo. There was a well-established protocol. I said, give me the biggest dose. It was very intense. I needed 4 blood transfusions just to keep alive. I also did one other thing, and that originated kind of this, that it was coincidentally that the same year I got my tumor, there was a Nobel Prize in chemistry for something called click chemistry. It's a really cool thing they discovered where you can have a reaction in humans that only happens, only the reaction you want actually happens. It's an amazing technology. It won a Nobel Prize for a reason. But the guy who's first in humans with that was in my YC batch. We became best friends. And over time, I became his biggest investor because before the Nobel Prize, no VC believed in it. Biotech VCs are incredibly hard to raise from because biotech is very expensive, and they only want to do consensus stuff. So it ended up becoming my biggest investment, and now that I had cancer, he offered to also make me a patient, which was amazing, and which taught me a lot about what was possible in the system. You could do a trial for a single person, a so-called single-person IND. Despite all of our efforts, 2 years later, we saw local progression. And my oncologist said, hey, I can't recommend any drug for you right now. Maybe there's a trial. There was no trial. I have a relatively rare disease. So it was a real oh shit moment. I quit my day job as CEO, and I started focusing on this full time. And me and my wife went everywhere. We talked to everyone. We're still doing that. And we went to medical conferences. We did a tour of like all the academic hospitals in the US, it feels like. We talked to every doctor we could talk to. And we went everywhere. This is a picture of me last October doing an experimental treatment in Beijing. Oh, it's a diagnostic, not a treatment. But we settled on doing 4 things. We're going to do every diagnostic that remotely makes sense. We're not going to know in advance whether it will be helpful, but we'll just do it. we're going to make our own treatments. If we're out of treatments, let's make them. We've seen it before. It can be done. It takes time. We're just going to do this. And we're going to do everything in parallel. We're already kind of low on time. It could metastasize at any time. Most people run out of time. And let's do as much as possible in parallel. And last but not least, I like starting companies. Let's try to make this accessible for other people too. Regarding the diagnostics, I did everything. Single cell, spatial, all the biomes you can imagine we've done. There's a lot of this online. So if you go to osteosarc.com, there's 25 terabytes of Google buckets. There's a bunch of online viewers. It also helps as a patient, if you ever become a patient, put your data online. People are intrigued. There's not a lot of public data there. It makes the getting conversations a lot easier. And we started our manufacturing campaigns. We did easy things like peptide vaccines. I've had 3 by now. We did things that are incredibly hard, like a CAR T. It's still like halfway through. And everything in between in how hard it is. And we did this with all kinds of people, with companies, with academics, you partner with whomever you can partner with to do this. I've had a doctor tell me to my face, "Hey, Sid, you're doing so much in parallel. If something cures you, we will not know what will have cured you," encouraging me to slow it down. That is bonkers. Like, you can do tests. It makes sense to do tests and studies. But if your only goal is to cure someone, just focus on that. And I think if you look at cancer care today, there's people are way too hesitant to do things in parallel. What doctors will tell you initially is like, oh, there's not a study that proves this combination is safe. Well, that's horseshit. It costs $2.7 billion to get a cancer drug approved. Do you think anybody's going to run trials for every combination of them? It'd be exponentially more expensive. It's not going to happen. I think with your oncologist, you should discuss first principles. If one thing hits the liver, one thing hits the kidney, maybe there's a chance to combine things. Most people run out of time. Your doctor is minimizing liability. You are maximizing survivability. Those are thank you. Completely different things. I didn't get this at all when I was dealing with doctors for 2 years. But they have a completely different incentive. They want to stay out of trouble. The hospital never got into trouble for someone dying of cancer. They did get into trouble for treating someone and having adverse effects. They are much more conservative. Even if you push doctors as hard as you possibly can, you're not going to end up with something that's bad for you. They will always be more conservative than is beneficial to you. And thanks for the applause. A lot of what I did is extremely expensive. I'm really lucky to have had a successful software company and be able to afford it. Some of what I'm doing, the price is coming down, especially the sequencing stuff. You can get your RNA sequenced for $50 at Plasmidsaurus. You can do whole genome for sometimes $500. AI is incredible. I don't have to tell anybody here, but it is really, really cool to see. You can just upload your raw RNA data, and it will say intelligent things about your cancer. The models are getting better all the time. Last week, we did a talk at the OpenAI Forum with a few more AI examples. It's incredible. And I think as a patient, you can do a lot with AI to advocate for yourself and make cases to your oncologist. And last but not least, the treatments in parallel, it could be a lot more expensive, but if it's a generic you're adding, typically it's affordable. And for some of these, you can get insurance to pay for it. Insurance paying is not a binary thing. It's also how loud you are, how many avenues you pursue. Of course, you're probably dealing with cancer, so you might be You might have a lot of other things to deal with. You might not have the energy. You might not have the time. A lot of people are saved by their partners. I was lucky to have a partner who stood right by me every step of the way. But advocate for yourself. Yeah, thanks, Karen. But one thing you cannot do, you cannot afford to make your oncologist responsible for your care. They are responsible for your treatment decisions, but they only have a very limited time to advocate for you. You need to be your biggest advocate. Your life is on the line. We started companies as we saw things where it didn't make sense that it didn't exist. So on the left, you see a few companies in diagnostics. For example, it's incredibly hard to get fresh tissue out of a hospital. If you have fresh tissue, you can do a lot of sequencing that you cannot do when it's FFPE'd. And typically, pathology will FFPE it unless you push back extremely hard. So we started a company just to get your own tissue out of the hospital undamaged. We started a company for diagnostics. We just started Relit. It's going to try to do diagnostic scans. Because every time you take a medicine, a lot of medicines target certain things. With a diagnostic scan, you can see how that pans out. I'll have an example later. We started a ton of drug companies. And all these companies are patient first. So they are financed not by some biotech VC, because those are extremely hard to convince, because they know it takes $2 billion to get a cancer drug approved. They're financed by the patients, because approval costs billions, but the prices for manufacturing something are getting down. They're coming down. It's like under $1 million, $1 million, $2 million. Most people can't afford that, but there are starting to become patients who can't afford that. And we'll start there and drive the cost down over time. I, as many of you here, care a lot about kind of regulations and making sure that regulations are in the interest of the citizens. And it is completely bonkers how the current system is structured. I understand where it came from, but we ended up with something that doesn't make sense. And I'm not an MD, I'm not a PhD, but you could just see that stuff is not as it should be. There's a lot of amazing people advocating for change, especially one I call out, Roxandra, who's doing clinical trial abundance. AI, for example, is making it easier to make medicines, but of biotech R&D, only 3% is spent on discovery. The rest is for clinical trials. And you want to do a trial? Great. Now you need the IRB of every hospital participating to sign off on that. Who's in the IRB? The people who really love reviewing your application and finding problems with it. What do they have to gain if they approve it? 0. What do they have to lose? When they make a mistake, the FDA pulls all clinical trials from that hospital. That's an extremely high bar, and it makes it really, really hard to get treatment. This is a lot of things. I think maybe people can call out stuff they want me to elaborate on. Just call out a number. 2 to 5. So 2 is phase 1, safety. It should be a notification. So right now, you want to start a new trial to test a new drug. You're going to be reviewed in any case by an IRB. They're going to look at everything. Then you still have to also go to the FDA, and you got to wait for their approval. That takes months. It rarely adds something. And in Australia, they say they replace that step with just tell the FDA instead of waiting for them, and they're just as safe. So it's useless time, and that time kills a lot of new medicines, because startups, the most important thing they have is time. So we could easily change that. Just adopt the Australian system that's just as safe, provenly so. Number 3, right to try. There's a lot of invisible graveyards of people who die because they ran out of medicines to try. And it is completely bonkers to tell someone, ethically, we don't want you to take this drug because it might kill you, when the alternative clearly is dying of cancer. I'd rather die from treatment than from cancer. Cancer is a horrible way to go. And most treatments are at least quicker. Market some drugs after phase 1 is complete. Right now, we say after phase 1, you got to do phase 2. After phase 2, you got to do phase 3. And only then you can suggest using the drug to people. For canines, we already say, hey, some drugs can be conditionally approved. And let's just follow the data and see how it does. It doesn't make it any more unsafe. We allow it for canines because it's the ethically right thing to do. But I think if my cat deserves it, I deserve it too. Canines are maybe dogs, but I'm not an MD. I told you that in the beginning.
So in the US, as you probably know, it's not possible for a patient to fully waive their right to sue a doctor for medical malpractice, even with a blanket liability waiver. I know here they rely on private arbitration agreements that at least cut out juries that make emotional decisions. Do you have other solutions, or do you think we just need to rewrite the laws so that patients can no longer sue doctors for experimental treatments? Is there a better way?
I don't have an opinion on that. I think it's really interesting. I think maybe even if you insert an arbitration agreement, I think the patient still has the right to sue in a regular court in front of a regular jury. I think even that you can bypass. It's a giant problem. Hospitals have such a high kind of cost in these lawsuits. They make a lot of money, but of their kind of incidentals, this is a really high expenditure, and every major academic hospital is focused on keeping these costs low, so they can't take any risk. Yeah, all these lawsuits, they lead to a very risk-averse thing from the major hospitals, and I think new jurisdictions could be a great way to improve that. And I agree, it would be great to do private arbitration and things like that there too.
Could you please expand on number 9 about the approved algorithms and processes? And would any of number 9 contradict what you're saying about number 10 for parallel treatments, especially in the situation where we might try let's say we try a molecule that's not approved. How can we try an algorithm that's not approved, especially if we need to verify identify what worked in order to get it approved?
Yeah, so number 9 is about, right now the FDA approves a molecule. Like, you are getting this molecule and you got to test that. Imagine you make like an mRNA vaccine. It's a different vaccine every time. And there's a lot of repetitive stuff there that you got to do every time. Instead, it'd be great to certify like, hey, we're going to make it in this way. We're going to use these ways to get to the vaccine. So these are our input data. We're going to run it through this AI model. This is our output. And then if you prove that in a phase 3 trial, then it's approved even if you put new input data in and you make unique new molecules. At least that's my hope with number 9. Number 10, the parallel treatments. We got to lower the bar from this has never been proven to work. It would be great if doctors felt the freedom to reason a bit more from first principles. We used AI, but also a pathologist in the loop to say, hey, look, are these side effects compounding or not? And if they're not compounding and you can kind of handle the 2 side effect profiles at the same time, you can combine medicine. And sometimes you kind of lower the dose of both a little bit. But the biggest problem with cancer treatments is that it escapes the treatment. If you hit it with multiple treatments, those odds are much lower. Like, suppose there's a kind of a 1 in a billion chance that there's a cell that can escape the treatment. If you have 100 billion cells, it's not going to work. If you do 3 treatments at the same time, the odds are way more in your favor.
I'm curious, actually, maybe Eric, you can talk a bit about that. So, I mean, here you can sign away liability, right? But then you might be contestable in other jurisdictions. So how would that practically work? And is there a potential to do something like a right to try haven here?
Good questions. Probably for a deeper dive of the legal team, but as it relates to all activities carried out within the jurisdiction following all the jurisdictional legal requirements, I do believe it can be a contained liability within the jurisdiction. But since I got the mic, Sid, I know you've not spent that much time here, but I think you've quickly absorbed what is happening and what could be happening. If at some point during your presentation you can talk about what we're doing here could enable a leapfrogging effect of sorts, given your personal experience and how you see the world in this field, it would be lovely to hear your perspective. It might already be aligned with what we're thinking, but nevertheless, it would be lovely to hear it. Thank you.
Yeah, Eric, thanks for that opportunity. When I arrived here, I went to the opening reception. I was kind of late, and the people from Unlimited Bio, I met them. I sat outside with the whole team, and it was just magical to hear them. They have found a way to make medicine much more affordable than anybody else, much more quickly. And then here in Prospera, they can dose. And they can get it to patients, and they can kind of legally export it from where they're getting it. They can dose it here. And I think that's the power of Prospera. And I think what a lot of the biotechs I talk to are looking forward to is having kind of an emergency department here, or at least the basics of that. As you build up the biotech infrastructure, you can do kind of more and more risky treatments that might have the cytokine release syndrome that Popvax talked about earlier. It's really exciting to see what's already possible with injections today, but as you build out the infrastructure, a lot more is possible, and that's the biggest thing holding people back. Discovery, design, AI can do it. Making it, there's labs who can do it. I think finding a dosing site that's responsible, that has a good IRB that Prospera already has, but then also has the medical backup to take care of people if there's an adverse event is very important, and you're building towards that, and that, I think that's incredibly exciting.
Maybe you can talk a bit more about, because you took a whole stack of different regulatory pathways. You mentioned a single patient, IND, you went to other jurisdictions like China, and if I recall correctly, might have also been Germany. There's also accelerated approval pathways. Can you kind of walk us through what you used and what was your experience with it? What works and what?
Let's talk to about some examples. So first, the good news. Now for 10 months, we have not been able to detect my cancer. And thank you. Not for a lack of trying, and we've gone from 0 treatments available to me to 30 treatments that I never all hoped to use. But we got options just by digging for them. I want to talk a little bit about what we think was very impactful. We did single-cell sequencing. In single-cell sequencing, you sequence a lot of cells, but you do that for each separately. And you can make a very colorful picture with all the different types of cells. So if you look at the purple there, those are my cancer cells. And you can zoom in on their genetic profile. And what we saw is that they had a lot of FAP, bottom right. FAP is fibrous tissue. Think of it as scar tissue. It's very common for osteosarcoma. And we found that there was an experimental therapy in Germany that targeted FAP. So first you do a scan in the morning of an FAP binder and gallium, where you can see where it lands. For me, that was amazing because it lit right up, right at the cancer site. So it was like, well, come back this afternoon because we're going to hit it. So they hit it, one time with an alpha emitter, one time with a beta emitter. You can see me here. I was in isolation for 2 days after. I had a real glow up, but I had no discernible side effects, but my cancer had. The cancer got 60% necrosis, 20% shrinkage. It detached from my dura, from my spinal column, and we were able to scoop it out via surgery. And we don't know what happened, as that doctor so famously said. We don't care that we don't know what happened. But what could have happened is that we really made the immune system really angry beforehand. We did dual checkpoint inhibitors. We did NK cell therapy, where you grow your natural killer cells outside of your body and place them back. We did an oncolytic virus. We did tons of things to rile up my immune system until it was ready and angry, but just didn't know what to hit. Because the problem is with solid tumors, they kind of hide. And in my case, we think that the FAP was signaling to the neutrophils to hide from the immune system. One thing was a virus that dropped the TGF-beta, but probably by killing the FAP cells, we stopped that signaling, and it kind of lost its invisibility cloak. And the riled up immune system that was the angry bouncer finally had the client to throw out, and it went all over that. And in the picture on the right, you can see that the T-cell infiltration in the tumor really grew a lot. Now, not all T-cell is alike, and it's not necessarily always a good thing, but in this case, it was a really good thing, and it was ready to take on what was left of the tumor after surgery. I also was able to do a personalized mRNA vaccine. In fact, on Monday, I'm scheduled to get dosed again. And it's really interesting how they make things like these. You have like these things on the vertical axis, all the different proteins you encode for. And then on the horizontal axis, you have all the different ways to compute that. Like, does it make sense? Is it present in the sample? What do the different models say about it? You kind of pick your targets. We're able to do that in 6 months. So we went from starting the project to being the first patient dosed in a new experimental trial at a hospital that had never done it before. This was in the US. This was including all the paperwork and everything else, but it still shows what's kind of possible if you push really, really hard, and when there's tons of data and expertise available. We do a lot of blood monitoring too. Every month, I take about 20 vials of blood, and it's sent to all kinds of different locations. If you want to see the signal from that, please go to osteosarc.com/timeline, and you can kind of follow the measurements that are happening. We also made a personalized TCRT. In fact, Willie, if you want to stand up. Willie's there in the back, in the blue shirt. He's going to be giving a demo tomorrow, and he's working on that TCRT. It's a very potent treatment, and there's lots of kind of single cell data, lots of sequencing that goes into designing it. By the way, this whole presentation is also available online if you want to see the pictures better. We also did functional screening. In this case, you take cancer cells, which you can do if you freeze it, if you have fresh tissue, and you grow it up in the lab and then test medicines against it. And it's not perfect, but in my case, we had a very surprising result. There's checkpoint inhibitors. The popular one is Pembro, and Pembro didn't really work, but a very similar one, Jemperli, or Starlimab, worked much better. We don't know why, but it's helpful to know, because then you can just use the other one, because they should be similar, but they aren't. And if one works better, great, let's do that. So this is the data on which we based that. We also found something that sounds like a party drug, but isn't. It's called MDM2, and I had a lot of MDM2. I am from the Netherlands, but I've never lived in Amsterdam. I was really, really high on the MDM2 scale. So I'm the index tumor here, like off the charts in expression. So we're like, okay, great. Who's made a drug? Like, can we get access to a drug? And multiple people tried to make a drug, and it has passed phase 1 clinical trials. But then they ran into the problem. And the problem is, if a drug only works on the minority of patients, it's uninvestable. You don't get money from the VCs to invest in that. It has to be a blockbuster. It has to be a GLP-1. It has to be at least a cancer drug that the majority of people can get. Because most people never do sequencing. So you're not able, the people who would benefit from your drug wouldn't even know how to find you. And then still, $2.7 billion for cancer drug approval is too much money if it's not the majority of people. So I believe a lot of the cancer drugs in the future are going to be financed by people who need them, and they're going to go 1 patient, 2 patient, 3 patient, instead of biotech VC money. That model doesn't scale to the number of drugs that we need. When we called the leading asset there, they said, oh, it's interesting that you call because we were about to throw away our multi-million-dollar drug supply next month. Do you want to pay for keeping the freezers on and the patents in protection? And it was $20K a month. So now we're paying $20K a month to keep the freezers on and hopefully rescue this drug. Because I think in the future, a lot more people will do the genetic testing and there will be a market. One thing we're doing is a CAR T program. It's a very potent but also very tough to design drug. We choose B7H3 as a target. It's some kind of target. People are excited about it. But we found that in China, you could do a scan against that. It was the first place in the world where we were able to get access to a scan for that. So we flew to Beijing and became very fascinated with the speed of China. I went in and out of that academic hospital in 2 hours with the scan results in hand, which is incredibly fast compared to the US. And they had good news and bad news. Good news, they didn't see any cancer. Bad news, my liver lit up 3.5 times more than the 20 Chinese people that did the scan before me. So we went back to the CAR T maker and said, hey, Cole, can you do something about this? He said, well, actually, I published on snipers, a logic gate, an AND gate, where we can have 2 proteins show up before it activates. So now he's adding an additional logic gate, an additional protein that has to be present. It's FAP in my case, because we know that's expressed, and it will probably still be expressed. And I think it's really, really cool that you can do an experimental scan and then take the result back to change how a medicine is developed. And I think these radio diagnostic scans have a big future ahead of them. Last story, we saw something highly expressed. It's called PANX3. It's very unknown. It was like 10,000 times more in my tumor than anywhere else. We're like, oh, this is a nice target. Why hasn't anybody done anything with it? And we found out it's hydrophobic. So if you normally test for targets, you're going to do that in a water-based environment. It's hydrophobic. It will never show in your tests. So we think that's one of the reasons that people have never found it. We are running a binder campaign against it. We just concluded that it might be too expensive to make a medicine with it in the US. But then I had breakfast today with the people from PopVax, and now we're talking, so we'll see. So thank you all. This is an amazing community. Thanks for listening to my story, and happy to take more questions.
Well,
great talk. Hopefully, a bunch of these N of 1 experiments are happening and continue to happen. Given that this is happening, how can we maximize the scientific value of self-experimentation?
That's a great question. I think one thing you can do is publish about it. Publish your cancer online. You know, it doesn't matter if someone steals it. Like every N of 1, it helps inform other people, including like what you did, how you did it. It will hopefully encourage more people. So I think being public about it is one thing. And then every time we do something, we can see the price coming down. These companies are started, most of them are pretty expensive, but it's like, it's the Roadster. And as we make more, hopefully we can get to the Model X and the Model 3. We've already seen that in the mRNA vaccine that went from 4 million to 10x less. So hopefully more will follow. Any other questions?
Hey, great talk, man. Thank you very much. The piece around taking live tissue and then being able to grow it out in a lab, could you elaborate a little bit more on that? Because that sounded like the most fascinating piece of this. And like, could you just give it a little bit more detail on that?
Yeah. If you're able to have fresh tissue, which is very hard to save it from the claws of pathology, but if you're able to, you can send it to providers like Travera, and you can test against it. You can test it with kind of chemo drugs, but you can also test immunodrugs, which is incredible. So these checkpoint inhibitors, for example. There's also growing bigger tissue. So allosoracne, for example, does that, where you create a real 3D structure, which is more representative. And if you're lucky, and we haven't been lucky, but we've tried, you can make a cell line that just keeps going, so you don't have to do any new biopsies, and you can keep testing medicines in the future.
Hey, thank you for the presentation. I'm very familiar with GitLab, Darren Murph, and everybody. I was in the running outside, and it's incredible to see kind of this second, third life of yours. I'm very curious on how you went from leading a software company with an IPO, which was incredible, to now going full founder mode with 10 companies. What were the transferable skills that allow you to do this leap from one market to something completely different, and maybe can be inspiration for many of us that are always changing markets, especially now with AI?
Yeah, thanks for those kind words. It certainly feels like a second life. I think the transferable skill is kind of a bit organizing people, but also kind of the Silicon Valley startup mentality of you make it a separate company, you give it a CEO. The CEO has only a very limited set of things to do. None of these companies have like 10 drugs or a complex pipeline like that. You have a very clear focus. And then we're not going to we'll develop it for 10 years and hope we have a breakthrough. No, you're going to get patients in year one. You're going to make it for someone. And making it for someone speeds things up so much. It's hard to describe, but everything becomes easier. People start thinking differently, and people have different pathways, like the single patient IND, which is only available if you have an actual patient who is in need of the drug right now.
Hello. You said one thing that made me think about something. You said publish your diagnosis, or if, God forbid, somebody has it, they should publish it. And at the same time, when I looked at the presentation, it seemed that access to information about what exists out there is very important in order to build all of this. So I'm thinking, because it's so expensive, is it possible to create, at least for some cases, a sort of a bounty or a hackathon that opens sources and crowdsources information and then attracts people to compete to solve, based on that information, the problem, and then maybe use those 10 companies as a layer of that solution? But I don't know, this is just like, it just occurred to me, so I don't even know if this makes any sense.
Yeah, thanks for that. It crossed my mind, hey, could we pay people for opening up their information? Because there's a lot of startups that want genetic data, and right now they have to get it one at a time by paying a lot per sample. It should just be in the public domain for everybody to train on. So could we pay patients to open it up? That would be one thing. The other thing is, as a patient, as you open it up, should you pay people to should you organize a competition? Like best designed mRNA vaccine gets $1,000. I think most companies won't actually go for the $1,000. If you just say, hey, I'm in need of an mRNA vaccine and I got manufacturing lined up, there are companies that will help you. People are now kind of finding me, and every month we have a presentation from another company kind of doing mRNA design. And we found like Serova, who is doing an amazing job at mRNA vaccine design. We would never have found it if I wouldn't have opened up my data.
Hi there. Similar idea on the incentives piece. Just interested in how much have you actually spent on the whole treatments that you've done and all the research versus, say, standard of care, which is maybe are less effective but could actually be more expensive, like just to kind of understand where there might be an incentive for the health insurers or employers or whoever might be paying for your health care to actually support the funding of treatments. Because as you say, the manufacturing is often a lot cheaper than the approved drugs. And if you're happy to actually try an experimental treatment, it might be cheaper than standard of care.
So my standard of care was way cheaper than manufacturing the medicines. I think it's a best case where manufacturing a medicine is cheaper than the most expensive drug. I think it's true for a few, but the vast majority, the first patient getting something, it's way more expensive, order of magnitude, 2 orders of magnitude. So for now, it tends to be privately funded. There are a few things you can do, like paralyzing generics and things like that, but actual medicine manufacturing, it's just starting to come into the realm where a few people can afford it. But we'll bring the cost down quickly so more and more people can benefit.
Hey, Sid. Good to see you. Congratulations. Amazing journey.
Thank you.
So say you had access to a novel jurisdiction where you could accelerate some of these things. What would you suggest to companies or to people that had influence in that jurisdiction that they do first to accelerate something like this?
I think it's incredibly hard and complex, and I think it should be a conversation and not podium wisdom. But it seems that manufacturing tends to be easier than actually the injection side. So the biggest need is like, how good is the injection side? Povvax talked about cytokine release syndrome. I saw the clinic here today. We can't have someone there with cytokine release syndrome. So like getting that up to emergency care, to intensive care, getting CT scanners, MRI scanners, breathing tubes, the whole thing, like every step we can make there, it's going to unlock more. I think that's a really important thing. And of course, all the work that Nicholas is doing, getting US jurisdictions to open up is also incredibly important.
I'm curious if what role, if any, did insurance play in helping you along this path, or was it all out of pocket? And was it complicated by the international scope of your journey?
So thank you. So poor Cigna, they're calling me like every week that they would like to assign a case manager on my case. I don't think that's for me to access more treatments. I think that's to reduce their bills. So I'm not open to that. I think a lot of what I did, like the experimental treatment in Germany, it was not covered by my U.S. health insurance, but it was something that most people in this room could afford. So there's a lot of things that are very expensive of what I did, but there are things that are within reach. But then there's other barriers to access. Just getting in touch with people, language barriers, walking around a medical campus in Germany from the 7 different offices to find the right spot. Like there's lots of little things along the way, but yeah, a lot of these things insurance won't pay for, and like insurance only have that much money. I'm not advocating they pay for more.
Yeah, thank you, Sid, for being as open as it gets about your case. I feel like it deserves a 3-day conference of its own just for us to understand how those findings can be applied to modern healthcare. And the question is, say if you are like a regular person, maybe you have like 100K to 100K saved up and you have, say, a similar case to yours, what would be the best approach to spend this money?
I think getting fresh tissue, having that sequence, spatial and single cell, and then feeding that through AI, and then talking with as many doctors as you can, and putting a lot in travel cost, and then saving a little bit for something in Germany or something in China, and then trying to take advantages of many generics and putting a little bit of pressure on your healthcare provider to pay for things. I think that's what I would focus on.
Sid, I think I'm speaking for all of us. We're very grateful that you're here today with us due to your own entrepreneurial effort, ingenuity, and also the science and technology that was making it possible. Your case is a huge inspiration for all of us. Thank you.
Thank you so much.